So I'll start. Good afternoon to our panelists and to all the delegates attending the session. We have just an hour for this session, and we have 5 speakers who have about 10 minutes to introduce their topics. I have to apologize upfront that the time for discussion will be very tight. In terms of the presentation, we will start with Ashley Hayes, followed by Peter Durkin, and Professor Leslie Scott is replacing Prof. Wendy Stevens, Prof. Tulio de Oliveira, and Daniel Ndima. I will not go through the CVs of our panel members. Those are available in the program. My name is Sibongile Gumbi, and I'll be facilitating this session. Just as a way of introducing this topic, as Africa responds to the COVID-19 pandemic, we have experienced a number of technologies being developed in the continent. Countries such as Senegal, Nigeria, South Africa, and so on have supported local manufacturers to develop not only locally relevant diagnostics, but those that also have a global reach and will be globally competitive. These local developments have demonstrated that there exists value chains in Africa to develop the various inputs required for some of the in vitro diagnostic tests. In the management of any disease, early disease detection is absolutely critical. Thus, these in vitro tests, including point-of-care diagnostics, are exceedingly important. The market opportunities for diagnostic presents a sector that is viable. With limited barriers of entry, this sector proves to be growing in the continent. In this session, you will hear from developers who are not only developing on COVID-19 diagnostics, but are using platform technologies that have a far wider reach, market opportunity, and will be of impact. It is clear that platform technologies present the most viable solutions, enabling manufacturers to pivot their technologies for other applications. I won't take up much more of your time, and if we could move into our first panelist, Ashley Hayes. Thank you.
Yes, hi, good morning, everybody. I'll just start my video and share my presentation. Okay. Okay, there we go. Yeah. My presentation today is about medical diagnostic.
Hello, Nana.
Are you there?
Yeah, I am in the session.
Okay. My presentation today is about medical diagnostic.
I am in the online session. Yeah. I will come to the ICC tomorrow. Yeah.
Apologies,
because I was worried about this protest.
Can you perhaps mute your mic? Okay, there we go. Apologies. My presentation today is about Medical Diagnostic, a company I established in 2010. And Medical Diagnostic was established as a developer and manufacturer of rapid diagnostic test kits. I'm a biotechnologist myself, and I've worked on the methodologies to develop cheap, cost-effective rapid tests that can be used at the point of care. Our products are robust and can withstand extreme conditions. We've got SAHPRA approval on our test kits. We're currently going through validation to get a SAHPRA approval on our COVID products. But we have ISO 13485 accreditation. We've got a SAHPRA medical device license. We've also have WHO, CE marking, FDA on certain products. Recently, we've developed a fuel marker to mark petroleum and diesel using cannabinoids from the cannabis plant. So we've been also exploring biomarkers. We've got a team of experienced scientists heading up research and development. Some of our achievements over the years that we've achieved, Forbes recognized the company, of course, that have opened a lot of business opportunities for us. Also UWC, I'm an old UWC graduate, very proud of my old university. Then the products. So of course, in Africa, you need products that can be used at the point of care. And for that reason, we developed methodologies whereby you can test an individual using a blood sample, saliva sample, or urine sample, and you can obtain the results between 5 and 30 minutes. The test, of course, should also be able to withstand the harsh conditions in Africa. We've done a lot of stability studies to ensure that our assays can withstand 40 degrees for up to 2 years. And like I mentioned, we've got an ISO 13485 medical device license, and some of the other accreditations I mentioned earlier. Products that we manufacture kits for dengue, like I explained earlier, also for the motor fuel and diesel to check for paraffin adulteration, but also to see if there's a presence of biomarkers for traceability purposes. We manufacture drug tests, HIV kits, malaria, pregnancy, ovulation, schisto, and syphilis. The schisto test is the first urine test that we are aware of that is currently has FDA accreditation. We've developed this kit with the university in the Netherlands, and it's been distributed by a company called ICT Diagnostics. The sector we operate in, the medical device research and development sector, manufacturing and sales. We've got 40 employees. We can manufacture about 20 million test kits per annum. And also we own 51% of a company called Oculus ID. Current research and development projects, pending regulatory approval. We've got a COVID antibody IgG/IgM test, very standard test. We also have a neutralizing antibody test, of course, to check the efficacy of vaccines. And then also where we are with the technology, we're sending it to the NHLS for validation, and post-validation, hopefully we get our approval. So we do have different COVID products. We've got an antigen saliva test. We also now recently developed one for the nasal swab rapid test. It's performing quite well. We've done validations against PCR, of course, everything developed in Cape Town, South Africa. And we can actually detect as low as 0.25 nanograms per mole, which is quite sensitive. And it's comparative at this stage, very close to PCR. What we've also worked on, as mentioned earlier, we've developed a THC test to detect cannabinoids in petroleum. The mere reason for this is because the current markers could be adulterated out, or it could be distilled out, and the underworld also got their own methodologies of getting rid of it. What we've then discovered was that cannabinoids in the THC, in the cannabis plant, of course, cannot be adulterated out. So we've got a lot of research we've done on that. We've got external validation reports, and that, of course, is massive if it comes to marking fossil fuels. Then we also have a product called ParaDNA with a company called Genomics. It's a point-of-care thermocycler. You can actually do PCR in the field with it. It's the device on the left. And that is also going through SAHPRA approval at this stage. We've done a lot of validations of the technology itself, and it works quite well. What we've also recently validated and intend on taking to market within the next 6 months is a product called Oculus ID. Oculus ID utilizes the camera. We actually intended on porting this to cell phones, but at the time, the resolution of cameras weren't good enough, and you needed infrared light. Now, these days with the technology, we've managed to successfully develop an Oculus device. It's patented, and what it does is it scans the pupils of the eyes, and it basically, a flash goes off, it then plots a graph, and based on the reaction of the pupil, it can then diagnose stuff like diabetes, but also substance abuse. So we're quite excited about this technology. We do believe that it could play a critical role in diagnostics and first point-of-care screening. So those are the algorithms running. The technology was developed in Cape Town as well. We published the PCT patent for this. And yeah, that's my presentation on different types of diagnostic platforms that we as Medical Diagnostics recently developed and intend on taking to market. Hopefully soon we get a SAHPRA approval on the COVID products and can assist in this fight. Thank you.
Thank you, Ashley, for that informative presentation. Exciting stuff that you are doing out there in Cape Town. And I think what's also very exciting is that you're actually manufacturing the products yourselves within the country. And I think that's a very good way to start with what you're doing. I will then move on to our next speaker, Peter Durkin, Dr. Peter Durkin.
Thank you. I'm just going to share my screen. Okay, is everybody able to see my screen fine? Okay. Right, so I am Dr. Peter Durkin. I'm the Chief Scientific Officer of Afrobodies. So Afrobodies is a South African-based biotech company, and we specialize in the production of recombinant alpaca antibodies. These antibodies are more commonly known as nanobodies. And there are two main parts to our business. One is the development and production of custom nanobodies for clients. And the second part of our business is where we develop our own nanobodies for projects of interest to our company. Nanobodies themselves are small biological molecules. They're only approximately 15 kilodaltons in size. This size means that they're about 10 times smaller than antibodies produced by other mammalian species such as humans and mice. Yet nanobodies are able to maintain equal, if not superior, antigen binding properties. Nanobodies themselves, they can be produced in a wide variety of platforms, such as in bacteria or yeast, plants, or in mammalian cells either. Once produced, nanobodies, they display excellent thermal stability properties, such as many nanobodies, you can heat them up to 50 degrees Celsius or more, and they still maintain their antigen binding capability. However, the most beneficial properties of nanobodies is in their adaptability. With nanobodies, you can attach a wide variety of different tags onto them, and this means that they become highly suitable for incorporation into a wide variety of diagnostic platforms. And I'll just show some examples of how nanobodies are being used by the diagnostic sector. So nanobodies have been used to develop highly sensitive point-of-care detection for HIV via the detection of the P24 antigen. They've also been used to detect animal pathogens, so for diseases of livestock. And thirdly, and most excitingly for us, is nanobodies have been attached onto microchip sensors. And they've been shown to be highly sensitive for detection of a wide variety of different molecules. The microchip sensors is definitely where we see the future for nanobodies and diagnostics, and it's the area where we're putting a lot of our focus on going forward. I'll now just briefly talk about one of the projects that Afrobodies is currently working on. So we are working on what's called a Companion Diagnostic for COVID-19. So for Companion Diagnostic, it combines a therapeutic element with the corresponding diagnostic. Our company has developed a range of nanobodies that target the spike protein of the SARS-CoV-2 virus. And we've had our nanobodies validated by scientists in the UK and also here in South Africa. And they've shown that our nanobodies are highly effective at neutralizing multiple variants of the virus. So based on these results, then we decided that we're going to incorporate these nanobodies into a companion diagnostic. So how is it going to work? So it will work like this, basically. So for the therapeutic pillar of the companion diagnostics, we're incorporating our nanobodies into a nebulizer-based treatment format. So we decided to focus on this treatment format because inhalation of the nanobodies directly into the lungs will allow quick and efficient neutralization of the virus there and thus stop virus propagation. The small size and stability of the nanobodies make them ideal candidates for this. And as a company, we're quite excited because we believe this will be the first ever African-developed immunotherapy that will be brought to market. On the diagnostic pillar side of the companion diagnostic, some of the same nanobodies that are in our therapeutic will form the platform of our rapid point-of-care diagnostic, which will be a microchip sensor-based option. So how it will work is that the patient will turn up to the GP's office. The GP during the consult will take a sample from the patient. He'll run it on our microchip point-of-care test device. And if there's a positive result at the end of the consult, then the GP will be able to prescribe the nebulizer-based therapeutic that contains the nanobodies we've developed, because he or she will know that the nanobodies in our therapeutic will neutralize the virus that's present in that specific patient. So in addition to this, we're also in talks with health departments about how we can link our nebulizer-based treatments to be accessed by close contacts of an infected person. So we want the close contacts to be able to access the nebulizer treatment as well, so that this will act as a type of prophylactic. And yes, we're hoping that this might make a contribution to help the world get back to a sense of normalcy. Thank you.
Thank you very much, Peter, for that very informative and a brief presentation. I think what's quite exciting is how you incorporating other digital technologies to actually advance the usability and utility of your own diagnostics. I think as we move into incorporating other technologies, provides for some very exciting avenues into other areas, such as now starting to incorporate artificial intelligence, starting to be more informative and informed about how to improve and to progress some of our diagnostics. Professor Scott?
Great, thanks. And good to be here. Apologies from Wendy Stevens, who's unable to attend. She had an emergency to address, but I'll be able to deliver the presentation to you. And I'm also the Head of Research and Development in the Department of Molecular Medicine and Hematology based at WITS, and also work closely with Wendy on the iLead program, which is Innovations in Laboratory Engineered Accelerated Diagnostics. So I think from the two previous speakers are more delivery on diagnostics and innovations in actual products, whereas our group really sits in the area of end users and implementation of diagnostics. So if you really have to look at what are the things that are needed for delivering quality diagnostics, we have to ensure that we have a good understanding of the needs across the pathology value chain, and what are the clinical algorithms, how will the tests be used, as well as a good understanding of the current landscape and the diagnostic footprint, so that you can better understand the context of where a diagnostic or a product will be used. And of course, to not forget the performance evaluation and regulatory environment. Somewhat difficult to navigate if you are a first-time user, but really to ensure that improved quality and ongoing standards are maintained within the field. So just a little bit more about the innovation opportunities. And when we think about diagnostics, we shouldn't really just think about a test or a platform, but really it's everything that encompasses the patient, the specimen, the transport of that specimen to the central laboratory for testing, as well as getting that result back to the patient and ensuring linkage to care through a system-wide connectivity. So if you look at some of the examples that we've mentioned here on areas, such as an improved specimen collection device, an improved specimen stability during the transport, making use of multi-purpose platforms in the laboratory so that you can use technologies across different diseases, And of course, not to forget the really transformative area of digital health technologies and the impact that that has on delivering diagnostics, monitoring diagnostics across the pathology value chain, and also bringing with it accountability in the various steps of the use of the diagnostics. So I just wanted to share with you some of the successful innovations and the opportunities that we've developed for products in the HIV and the TB environment, which is really our background, and had to rapidly transform it into COVID diagnostics. So these are some of the examples that our group have worked with, simple collection devices for HIV viral load testing, the TB spot, the dried culture spot, which was actually a gap in African Innovation Award and developed here in South Africa. It's now a quality product used in 27 countries to ensure diagnostics and their use at point of care and centralized labs. The eLabs digital health program really monitors accountability across the pathology value chain for viral loads, was rapidly transferred onto COVID and soon to be for TB. This monitors every quality step of taking the specimen and getting the result back to the patient. The endless areas that we see in innovations around big diagnostics, big data diagnostics, simple readers that improve quality of RDTs, and of course the really exciting area of the new TB diagnostic platforms that have been WHO approved and about to enter markets. So what are these innovations for diagnostics that we've really been involved in for COVID? Well, you can see that not only the rapid clinical trials and use case models and the performance in the field that we had to do and still do, but there's been really great innovations around developing our own reference materials, whether it's cultures, whether it's biomimetics, recombinant proteins that have come from labs within South Africa, stable RNA. These are helping to validate technologies and be a standard product across all different types of COVID assays. But there's a lot that goes into it. And Tulio will talk a bit further about the impact of variants and what they could have, in this case, on diagnostics. And I mentioned ramping up the digital technology area to impact on diagnostics. And you can even see a tiny diagram there of swabs. One swab's not the same as another. We'd have to validate that, check which ones have improved performance, improved stability, and which are best. Can we even use these for self-collection and self-monitoring? So in the digital era, this is really here to stay and really to expand. There are so many areas where digital tech can really impact on diagnostics, where the diagnostic can be the center, but then be able to provide the results directly back to patients from biometrics, wearable devices. Patients may be able to have their levels checked while they're wearing a device. And really, I can't stress more the need and importance of the digital era for diagnostics. So what does all this mean, though, when you come to looking at scaling and implementation? You really need to know the context. And this is in an era where, as you can see, I've shown the molecular diagnostics market is going to be worth billions by 2026. But without getting it into the field and without implementing in the right environment, it's going to be very difficult. So here's just an example of the National Health Laboratory Services National Priority Program platforms that are available for diagnostics in the public sector. 16 viral load laboratories can really generate more than 5 million tests of viral load tests per annum. The GeneXpert testing platform, more than 2.5 million tests per annum. These are platforms that could rapidly be leveraged for COVID testing. But if you look, the availability of these tests were not always there, not at the beginning of COVID, but something like the Allplex or the Thermo Fisher platforms. These are run on open platforms, and 46% of them are currently being used. This is a huge opportunity for diagnostics to be developed and implemented on these technologies. And of course, where are we going into the future? We need to look at point of care testing, which COVID has certainly shown us that that's what's needed. Digital pathology and NGS for cancer. There are endless opportunities for scaled implementation, and the basis is a good diagnostic. But it really needs to cover the full spectrum. We in South Africa have a hybrid model of very highly centralized laboratories, all the way through to district level laboratories. But in the COVID era, we pushed this even to mobile vehicles, being able to provide point of care testing or even specimen collection out in rural areas. This is where diagnostics have to really cover the full spectrum. But there are complexities, and I'm sure that those that are on the call and have presented have really witnessed some of the complexities and challenges, particularly across the pathology value chain for COVID. This is a very well known graph from Lomasa, who've really shown the disease progression of a COVID-infected individual. But what's not really well understood is the thresholds of detection for both serology and antigen and PCR technologies. And what I liked about this presentation of this graph is really to show the windows of the false negatives. There are limits of detection, limits of sensitivity and specificity for diagnostics. And I think that is really not well understood by many developers as to what's needed, when it's required, and where you can perform the test. So if you look at the pathology value chain, in particular to COVID, there are complexities that diagnostics are faced with. Specimen collection and stability can be a nasal swab or a pharyngeal swab, saliva. These all bring with it variabilities on the actual endpoint result. The process workflows is tricky depending on the specimen type. There are various platforms and gene targets in assays. And of course, access and availability is really key. So in our particular group, we've evaluated and worked with diagnostic developers as well as help provide knowledge to SAPRA for the robust evaluation protocols. And you can see in these funnels for the different types of assays for COVID, a large input, but very few output, and really quite tricky acceptance criteria that actually had to be developed way before guidelines were available. South Africa reported their first case of COVID in March 2020. Guidelines for being able to help us decide which assays are sensitive enough and which have good performance or not only became available from WHO in August 2020. So what happened in that gap? We had to develop our own protocols, our own materials, and our own ways of evaluating. But you can see here, specifically for the molecular assays, that we've learned a lot by being able to evaluate a huge number of technologies. We've been able to also provide information to developers to highlight which are the most well-used targets, which are the better targets in the highly performing assays, whether they are available for closed systems or open systems. So it's not only on the development side of the diagnostics, but also on the evaluation side that has lent itself to lessons learned, innovative approaches, and certainly providing knowledge. So this is not anything new to those coming to the session today, but really the importance of the regulation cannot be stressed more. We know that SAPRA has a very detailed navigation protocol. We know that they follow the reliance model, and really it is quite a tricky process and quite daunting if you have to start at the beginning where you submit your application. But as the process, and specifically for COVID, has really helped to mature this process. So from our particular aspect of working with companies to evaluate technologies, We undergo technical review with the groups. We select technologies with SAPRA, perform evaluations in either one of 3 phases, all the way through to clinical trials. Performance criteria are then reviewed, and all along the use of target product profiles for SAPRA licensing. But it's not only on the development of diagnostics and the companies that actually stress throughout the process of approvals, but also on the research diagnostic side. This is just a 13-step quick view to show you that there are a lot of approval processes that that need to go through for a particular diagnostic to be registered, to even perform a clinical trial. And so it really comes down to large efforts that I think need to be looked at between the different groups that overlap on their protocols and requirements. And I think something that can be reviewed and you know, made a little bit more speedy. So really, in summary, and some take-home messages for diagnostic developers and implementers, is that innovation is needed across the pathology value chain. It's not just individual products, but there are several along each step of the way. Processes and digital innovations really can drive diagnostics. COVID has pushed the boundaries to point-of-care testing and now even self-testing. Diagnostics have a huge potential, but they really are competitive. How do companies in South Africa go against the Roches and the Abbotts? It's really tricky and very competitive. But scale is everything. And the feasibility, you could have a niche product or products in large quantities. But there could also be a lot of knowledge transfer to South African developers. One of the areas we feel is important is also to involve the end user early in the development pipeline. Having a reagent is only one way, is only halfway to actually getting a test to a patient. As I mentioned, the regulatory environment is maturing, but does need some balance. And post-market surveillance is really key. There needs to be ongoing monitoring from the field. The importance of partnerships cannot be stressed more, but also the right partners. And I think the platform that BioAfrica have is really great because you can see the needs across the development to manufacturers to regulation. And I think partners can certainly share their experiences. We need to strengthen the knowledge dissemination through these networks. And of course, more importantly, is think beyond COVID to other diagnostics, repurpose what we have already learned and developed. And it's just to say thanks to a multidisciplinary team that really works across the pathology value chain. And that to just stress that, you know, it's fantastic to see developers doing their own innovations and being able to develop products. But there really is a huge amount of work that needs to go into actually delivering this to a patient in the field. So, and of course, thanks to all our partners and funders. And I'll hand back to you. Thank you.
Thank you, Prof. Scott. I couldn't agree more with your last point of thinking beyond COVID. And I think the challenges of other disease areas are ever present. Can we move on to Prof. Tulio?
Yes, good day, colleagues, good day, participants of the BioAfrica. Yeah, it's a great pleasure to be here. And I have been many BioAfricas before, and tomorrow I'm going to be going to the ICC and Wednesday to meet some people, but today, just to give you an online presentation. My presentation will be quite short, but we'll just highlight the big role that genomics have in these pandemics. Yeah, so I just call genomics, diagnostic, variants, and vaccines. Yeah. Why did I start with genomics? Yeah, because genomics has been potentially one of the most potent tools on the COVID-19 fight. It was used to discover the virus that caused the diseases. This means was used to discover through metagenomics, the SARS-CoV-2, very quick from the genome to develop diagnostics and the probes, was also used to develop vaccines. For example, both the new platform mRNA and vector vaccines really dependent of the genome of the virus to be put on them. Also to track transmission, to understand virus-host interaction, and to evaluate variants and vaccines. So it has been an incredible work across the world with 2.5 million genomes produced of SARS-CoV-2, never seen that scale, and over 15,000 of those in South Africa. Yeah. So what's the process that one uses to develop and evaluate, in the case diagnostic or genomics? And one start at the RNA of the virus, and then one can go to path once it's to test what Wesley was mentioned about qPCR detection and report. Now the method is really quick. You can do in a few hours, and some new methods coming very fast. But you can also go the path that you would transform the RNA on a reverse transcriptase PCR that's in the top. You would amplify, you would prepare the library, and then you would do sequence and analysis. And that's a massive developing on that area, and also in South Africa. And now we can go from sample to a whole genome within a couple of days. Yeah. So what we did in this pandemic, the first thing that we did is to set up a network, and that was incredible. We got 8 different universities working with 8 labs in the NHLS distributed across the whole country to do genomic surveillance in real time. First time that we come in such a large network, we call that the Network of Genomics Surveillance, that was funded by the Department of Science, Innovation and Medical Research Council, and allows South Africa to discover many things, and many very quick. For example, the first wave, we identify which lineages start the third wave, not only which lineages, but how they enter in South Africa and how they spread across the country. That was a major knowledge which could see, for example, strains that arrive here in Durban, is spreading very quick, or new introductions arrive in Johannesburg and going through the highway or the freeway to Durban. So we could really start tracking genomics, how the virus spread in South Africa in real time. On the second wave, many of you will be aware, we detected the first variant of concern in the world, which became then the Beta variant. We also helped the UK to identify their own variant, and Brazil. So South Africa really led on that area of very quick identification of these new variants. But the whole objective is not only identify, but really understanding at the protein structure what the mutations happen and what's the effect. So South Africa also did very well on that area. Once we have detected the variant, we have set up assays such as pseudovirus together with Penny-Moore at the NICD, or live virus that you could grow the variants in a very high secure laboratory, a P3+ lab, and then you could ask question, does the vaccines that are being trialed neutralize the Beta variant? And in the case here with the Oxford AstraZeneca, the answer is that large number of loss of neutralization. 3/4 of the virus tested could not be, the plasma from vaccinated could not neutralize this variant. And that was important and consistent about live virus and pseudovirus to determine which vaccines were used in South Africa. I did that together with a colleague of mine, Professor Salim Abdul-Karim. We organize all the data that have been produced both in the pseudovirus and the live viral plaque assay in South Africa, and allow to put an order of what would be the most effective vaccines to be used in South Africa, with, in the case, the Pfizer and the Johnson & Johnson having a lower decrease of neutralization and be effective against the Beta variant, and now against the Delta variant. So this is data that's been developed in real time in South Africa. What also allow South Africa and the labs in South Africa to become some of the biggest leaders on understanding the effects of vaccines on variants. So that's just a piece that came in Nature, highlighting how South Africa become one of the biggest players on that field. The NIH director, that's Frans Collen, highlighted South Africa as one of the examples in the world of the power of genomic surveillance to be used. And then the idea, what we are trying to do here through a network, is not only evaluate other vaccines or identify variants or publish big papers. The idea it is to try to have the tools in South Africa that we can challenge one of the big status quo, is that to start producing diagnostics and vaccines in South Africa. We have been very fortunate, a network of universities together with the Africa CDC, BioVac, and Afrigen, which I coordinate the research program, managed to win from the World Health Organization a big competition to set up the first COVID mRNA vaccine technology transfer hub in South Africa. And one of the main reasons to that, in addition of the companies, it was this very basic science that could take vaccines that are being developed and test that in real time, which both identifying variants, but also test the effect of the vaccines on neutralization, which is going to really help the country to try to become a player on the development of vaccines and diagnostics. So just to conclude, genomic surveillance is a critical component of the public health response. It really grew in importance with COVID, was used to identify the virus, develop diagnostic, vaccine, detect variants. South Africa has excelled in the genomics scientific research and received major investment from major funders around the world, which now we help to set up a genotype to phenotype in real time in South Africa, which is allowing the country to compete to become one of the first production hubs of mRNA vaccine. And this technology will also help to development and evaluation of diagnostics. Thank you very much. And just passing back to the chair of our session. Thank you.
Thank you, Tulio. I see we're starting to slightly run out of time. So I'll hand over immediately to Mr. Ndima from Cape Biotechnologies.
Thank you, Chair. And I think I will be very quick. I don't know if you can see my screen, right?
Yes, we can see it. Thank you.
Thank you so much. Thank you to you and my fellow panelists. I'm Daniel Ndima, the CEO of a biotech company that is based in St. Chiron, Pretoria. We are basically a company that was started as a project by the CSR through the support of the Department of Science and Innovation in around 2015. We launched this project in order to respond to, obviously, the shortage of reagents that we use in our healthcare responses, including diagnostics. And really, we launched the company as a private entity using private investments around 2018. But most importantly, we started really making inroads, in the US and formalizing some form of partnerships that are helping us to develop technologies that can really be competitive. But we also have key technology partners in South Africa that also include the CSIR. Our products now are in more than 10 countries, including Canada, US. Those are just key ones. And we are currently looking at 5 key markets in Europe. That will be German, French, Italy, UK, and Spain. So we really are seeing the uptake of our products. These products are sort of bioprospected from our indigenous biodiversity hotspots, the Cape Biosphere or the Cape Flora Kingdom. This is where the brand or our brand comes from, the Cape Bio Rich in Diversity comes from. We have developed a proprietary biodiscovery platform that allow us to be able to search for best-in-class enzymes, and we are able to use our proprietary biomanufacturing platform, which was licensed from the CSIR in order to produce these products at industrial scale. You know, our purpose as a company is basically to make sure that we present innovations that are coming out of Africa for Africa's benefit and beyond. I will be presenting a bit about our current platform. It's a reverse transcriptase quantitative polymerase chain reaction platform. I think Prof. Scott was presenting about some of the assays or kits that are developed using the same platform. We use these basically for RNA-based detection of viruses that have nucleic acid of RNA format via PCR route. And this platform is suitable for detecting other viral-related viruses. And this is where now we are able to expand the repertoire in terms of diagnostic development in South Africa. We have what we call a master mix or ready mix, which now propels us for development of a wide range of diagnostics that are based on RNA detection. Just to show an example of what I'm talking about here. So this is the COVID-19 sample that was analyzed in our laboratory just for the purpose of this presentation, just to show that we have seen quite a different performance, outlook of our product. That shows that any higher viral loads, maybe about 10,000 copies per mil, the CT values always come before the internal control, which is human RNA-seq, while the low copy numbers often come after this threshold, which our cutoff is around 37 of CT value. We also produced our own control, which is very important in a diagnostic landscape. What we have seen as well through interaction with the current customers, the end users of this product, is that they require that we package our products in master mix format so that there's a reduced workflow in terms of pipetting and all that. So we are able to customize accordingly to ensure that we don't disrupt the efficiency of the workflow, but instead we improve it significantly. So what this does is that it helps us, to have a lesser reliance on imports, as Prof. Scott was saying, that there's these big players in the industry that have been dominating for quite some time. But what this does is that it allows us to have a quick response to this outbreak on our continent, so that we don't really rely on foreign imports. But more so, it also saves us on our currencies. Being a local company means that we are closer to our clients. We are able to help them to optimize their protocols. We are able to help them, have access to these important reagents. But more so, what this platform does is that it has helped us to really create jobs that are, very desperately needed in the industry. And it helps us to now start to expand the value chains, and make sure that other role players are actually come through to assist to expand, the industry's capabilities. I think Prof. Tulia was speaking about some of these capabilities that they have initiated. So what we are doing basically is a complementing service to some of the work that Prof. Scott and Prof. Tulia was speaking about earlier. But what we have also developed is another platform that is DNA-based. We call it Cape Tech IVD. This is now, a diagnostic tool that can now detect DNA-based viruses or DNA-based detection. We're still optimizing this as well, and this platform can also be used in a DNA forensic applications. So the most important thing about the future of IVDs is that we tend to look at human benefits in terms of these tools, but we also have animals that one needs to take care of, including our own environment, in terms of making sure that we live in a healthy and safer environment. In terms of the market, I think there's a lot that was extrapolated from the previous presentations, but my interest here, or what I wanted to highlight here, is exactly what Prof. Scott was saying about the point of care diagnosis, which is a segment that is rapidly growing globally. And we can see that, in terms of the market share, this is starting to take off, quite rapidly. Though the molecular diagnostics is quite an important one, and we can see in terms of the growth rate, we also seeing that the point of care diagnostics are starting to take some form of shape. But what I want to highlight here as well is that what is driven the market is actually infectious diseases, and Africa is prone to quite unique sets of infectious diseases. And I think as a company, we would like to place ourselves together with our partners, including those that wants to partner with us, in order to expand the testing capabilities for the continent. What I'm trying to drive here is that, going forward, this process that you see here, where you have sample collection, I think Prof. Scott was speaking about it, nucleic acid extraction, amplification, etc. This is going to be integrated into what we call lab-on-chip point of care molecular diagnostics, where you will basically insert a sample. Inside that sample, basically, inside of device, you basically have the entire lab basically operating, and you are able to integrate that with a mobile device. Chair, I said I didn't want to waste your time, but let me close off here by saying that, when you look at IVDs in general, these are very critical and essential tools for making sure that we have adequate and safer vaccines and other therapeutics that are required for our healthcare. So if you want to be able to respond to this kind of crisis, one would look at ensuring that there's a platform that allows for multi-channel or multi-purpose detection of infectious diseases and among other testing capabilities. These are our partners, our investors, our partners in the US, Canada. We've got distributors locally. We work quite well with our government as we regard the projects that we undertake very vital for our local industry. Thank you very much. I think I will end there.
Thank you very much, Daniel, for enhancing and enlightening us. Certainly not time wasted at all. So thank you for the presentations of this afternoon. I didn't fully introduce myself at the beginning of the session. I'm the founder and CEO of a company called Smart Biotech, and part of the reason why I was invited by BioAfrica to facilitate this session is that we have just entered into agreement with Global Access Diagnostics that's based in the UK to collaborate on the supply, distribution, and most importantly, technology transfer of their COVID-19 antigen rapid diagnostic test in South Africa. This is a unique and exclusive partnership, as it not only looks at the access and availability of the latest COVID antigen RDT, but more importantly, that these diagnostics are affordable for the country. The COVID-19 test produced by Global Access Diagnostics, or GAD, is one of the best in the world for sensitivity and specificity, as well as providing fast and accurate results for wide-scale testing. So this is quite an exciting time and opportunity for us. We will start with just distributing finished product for an initial period, with a plan to back integrate through a technology transfer process towards manufacturing the diagnostics in South Africa. We believe this will bring a far more sustainable solution to managing the pandemic, as well as building manufacturing capacity and ensuring security of supply for the country. So I'd like to thank our panel members, especially for keeping their presentations nice and short and very informative despite the time limitations. We have had a balance of presentations, first from the diagnostic companies who are introducing and producing a wide range of relevant diagnostics. We also heard from research institutions involved in developing platform technologies, as well as supporting R&D work to improve the effectiveness of the diagnostic space through providing more information that we need. And I think this speaks to what Prof. Scott mentioned in one of her slides earlier about the innovation opportunities that exist, capturing the full value chain for an effective diagnostic landscape. I'm going to perhaps open up for questions. We don't have much time left. If we've got about 5 minutes, if there are any questions from the audience that's attending this session. I did invite questions in the chat. I'm not sure if there is any. Maybe to quickly pose a question first to those who are involved in the as entrepreneurs. How has the current environment and especially investments and resources that have been directed towards COVID, which has generated other opportunities, how has that had an impact in terms of business opportunities in your respective enterprises? And then for those who are largely involved in research, what has that meant from an R&D perspective in terms of the available resources, the available opportunities, and what are those kind of opportunities that have opened up for you recently? Thank you. Maybe start with Ashley, who started us off this afternoon.
Apologies. Can you repeat the question again?
The question actually is, given the recent, with COVID, there have been a lot of resources that have gone into R&D, into new business opportunities, new enterprising opportunities. Has the current environment in terms of the amount of resources allocated to COVID, has there been any spin-offs that you've experienced as a company as a result of the current environment that we're in, where there's a lot more alertness and awareness of the importance of new innovations for fighting diseases?
Yeah, definitely. Firstly, we got a grant fund from the Department of Science and Innovation via the SAMRC. So that assisted in developing some of the technologies. A limiting factor that I would say is the availability to access samples, actual clinical samples. That was a big hurdle for us to overcome. And we've managed to do clinical trials through a clinic. So the hospitals, however, assisted us in getting enough data to be able to submit to SAPRA now. So in a nutshell, in terms of funding availability, yes, there are funds available. In terms of spin-off opportunities and products in the diagnostic space, we had a surge in sales and other things. But of course, the limiting factor for us was accessibility to samples.
Thanks, Ashley. Any other responses?
Vangile, maybe I can just jump in here quickly from the research side. I must say that the recent months of COVID have really increased our efforts tremendously. And if I just reflect back, it took about 15 years for WHO to recognize the critical need of HIV viral load testing for monitoring of HIV. It took about 2 to 3 years for us to validate and get WHO input on gene expert testing for TB diagnostics. But it took 2 months for us to rapidly implement COVID diagnostics on similar platforms. So one thing that we have experienced is really the rapid rate at which things had to be done and the knowledge base that has really come from this. So I'm not saying pandemics are good, but it certainly has encouraged the research, has encouraged the R&D. And from the developers side, we've seen huge efforts on manufacture needs and innovation, and it just mustn't stop here.
Yeah.
Daniel, we've seen a lot of you in the media in the last 18 or so months. Do you have any comments?
Thanks, Chair. I wanted to also, have a few words here. I think, if your research project is strategically looking at, the future of the industry, or you position yourself quite strategically, and you have interactions with funders, and they're able to buy into your idea, and they understand exactly what you want to do, and they know that you are passionate about that, and you'll never stop until you make it happen. Investors look at the person and they marry those two with the technology. And I've seen that when we first went to the US and spent a couple of weeks, and we were just doing a talk to a few investors, and they were quite excited with the innovation that we're coming with. And you see that the US is mostly happy with the new thing. If you have something that is quite unique, with us, they were saying, for the first time, we see a technology like this, or enzymes that are produced out of Africa, leveraging on African indigenous microbial hotspots. This was quite a big thing for them, and they were happy to take us on. However, I have to say that locally, investment in R&D is quite scarce, especially coming from the private sector. But you'll see that the moment you have certain successes like we had recently of getting SAPRA approval, you start to see a lot of commercial investors coming through and saying, how can we help? And this is their opportunity. But R&D is quite a risky kind of puzzle. You may have 10 projects and then you find that none of them work. What do you do thereafter as a private investor? So that's where we need to have an integration between, interconnection between the private investment and the state, funding. And we are fortunate in this project that we had TIA, SAMRC, and DSI funding this project, but our investors were also coming through, and they put quite a substantial amount of funds to this project, helping this national response. So, the opportunities are there if you know exactly what you want to do, but you also need to make sure that you're able to navigate and leverage on other investments abroad, because there's so many people that wants to come and invest in Africa.
Thanks, Daniel. I think you've had an inspiring journey bringing Cape Bio to the fore. Peter Durkin, I wonder if you could answer, just maybe we need to close soon. There's this one question: what are the challenges in terms of accessing the right skills for diagnostic startups locally? Peter, maybe if I can direct that question to you.
Yeah. So by and large, the universities in South Africa do a pretty good job in training the people. So there is most, for our business anyway, all the skills that we need, by and large, I can source them here in South Africa. So that's a positive part of it. We're running into one of our biggest issues though is with the microchips. There's nobody in South Africa that can do this work, so I'm having to work with people abroad. But it is what it is, and as Daniel says, without investment, like you're talking like hundreds of millions of rands that really need to be invested to make a difference. So we'll see how it goes. Hopefully it will happen, and more of the private sector will come in on board to develop these things.
Thank you very much. It looks like we've run out of time. I'd really like to thank BioAfrica for putting this all together and putting such an amazing panel of members with diverse experience and coming from different areas, covering the full value chain as far as the diagnostic landscape is concerned. And to all those who have joined us for this session, it's been wonderful to have you join us, and I hope you found it as interesting and as enlightening as I have, and I've enjoyed chairing this session. So to everyone, thank you, and I hope you'll stay on to listen to more of this afternoon session. Have a good afternoon and good day to everybody.